Hello students, let's start the next class about cell signaling. As you have read before about the cell signaling in class 4, it's the class 5. So, in this class, we'll study about the RTK, receptor tyrosine kinase. What is receptor tyrosine kinase? It's a single pass transmembrane protein that have an intrinsic kinase activity in the C terminal that transport the phosphate group to the specific tyrosine residue. It has three parts, that is extracellular part, intrinsic part, and the intercellular part. Here, the primary signaling or the extracellular signaling that transfer into intracellular response without the help of G protein. It's normally act for growth, proliferation, differentiation, and survival. What are the key examples? That is EGFR, epidermal growth factor receptor, insulin receptor, PDGFR. Extracellular ligands and ligand specificity. Type of ligands that bind to the receptors are the fast messenger that have a growth factor or hormone cytokinins, that is that have EGF, insulin, FGF, PDGF, macrophages, colony stimulating factors, etc. It acts as a key and lock mechanisms where a specific structure like proteins bind to the specific area of receptors, then the receptor activates. After the binding of the receptor, the RTK change the conformations of the receptors and um, activate the receptors. Due to this conformation change and lock and key mechanisms, there is no accidental trigger of the massive cellular growth phases. Let's study about the structure. The extracellular structure that contain a highly specific binding pocket, as I said before, like key and lock mechanisms, the receptor bind to the extracellular domain. The the inter the transmembrane alpha helix structure have a uh, single transmembrane helix that anchor the receptor in the lipid bilayer, and the intracellular domain that have a tyrosine kinase domain, the catalytic engine, which which will be activated by transphosphorylations. The trans residues, tyrosine residues that present in the C terminal are being transphosphorylated to further activation of different secondary messengers. Here, how the receptor activate? When the ligand bind to the two RTK monomers, then uh, step 2 begins, where the step 2 start with the monomer side together and form the dimer after the receptor being bind to those two. After the binding of the two receptor and functioning as dimer, the transphosphorylations occur, where the first receptor activates the second receptor by cross-phosphorylations, and the second receptor activates the first receptor by cross-phosphorylations. After the phosphorylation occur, the phosphorylation sites act as a higher affinity docking site which recruit the SH2 domain. The RTKs signaling hub activate uh, different signaling as a multiple parallel cascades, where it activates RAS-MAP pathway, PI3K ATK pathway, mTOR pathway, PLC gamma pathway. Let's study about RAS-MAP pathway. In RAS-MAP pathway, after the activation of receptor and cross-phosphorilations, GRB bind to the phosphorylation site. GRB act as a GRB to act as a SH2 domain, and this SH2 domain then recruit the uh, SOS enzyme. That SOS enzyme is a guanine nucleotide exchange factor, as similar to the G protein receptor GPCR. Then the SOS force the RAS protein to be activate uh, by GDP to GTP. After the RAS activate, it creates the cascade of activations, that is RAF, MEK, and ERK. During the RAF, MAP kinase activates, then MAP MAP kinase. After the ERK activation, it enters the nucleus to activate the transcription factors, and the transcription factors activate and express the cell division and proliferation. Then about PI3K AKT pathway. After the receptor activate and cross-proliferation of the phosphate group in the direction site, it recruits the SH2 domain. The SH2 domain activate the PI3K, and that act on the membrane and phosphorize the PIP2 to convert it to PIP3. After the PIP3 activate, it acts as a chemical anchor and recruits the AKT kinase. After the AKT kinase recruit and phosphorylated and activate by other bounded kinase like PDK1, etc., it promotes the cell survival and anti-apoptotic signaling. Like it inhibits death protein like BET and caspase. The next talk about mTOR pathway. After the activation of AKT, it directly and indirectly activates mTOR. Basically, it is the targeted site for rapamycin. The mTOR has two types, that is mTOR C1 and mTOR C2. The mTOR C1 activates protein synthesis, metabolism, and cell growth, and the mTOR C2 activates the actin cytoskeleton and cell survival. What is the role in the cell? It dramatically increases the cell protein synthesis, promotes cell growth, and regulates cellular metabolism based on nutrient availability. Then about the PLC gamma pathway. In the PLC gamma pathway, the target directly recruit the PLC gamma, and the PLC gamma cleave PIP2 into DAC and IP3. The IP3 act as a secondary messenger and bind in the receptor of endoplasmic reticulum and release the calcium availability in the ER to the cytosol. After the release of calcium to the cytosol, it's bind to the C2 domain of the PKC protein and target it to the membrane. The DAC present in the membrane or membrane bounded DAC protein bind to the C1 domain and pull the auto-inhibitory pseudo-substrate out of the catalytic pocket where the PKC activate. Here is the summary of the RTK pathway and about different pathway, that is RASMAP, PI3K AKT, mTOR, PLC gamma. In single page, I have given the summary, so you should read and summarize all the things for the CSIR exam or ICMR GATE, etc. Thank you for the classes. Okay, have a nice day.